Hi, class. As you know, in-person classes are canceled for Friday, so I am here to give you your Chapter 4 Part 2 lecture that you can watch, listen to over the weekend, and be prepared for, hopefully, to meet back in class on Monday to do Part 3. I don't know if we will be back on Monday or not. I'm just hoping so. But yeah, let's look at your assignment. Okay, so you can already answer number 1 and number 2. You were able to answer that on part or after listening to part 1. With part 2, you'll actually be able to finish this lecture or finish your assignment. Or after part 2, yeah, finish your assignment. We're going to talk about the sliding filament theory today. So you're basically just outlining the steps kind of in your own words, but also taking a lot of what you see and hear from that lecture. We're going to describe what proprioception is and the difference between muscle spindles and Golgi tendon organs during exercise. So giving an example there, like during exercise, you're explaining the role of each one of these proprioceptors. And then your critical thinking is talking about fiber types. So we're going to learn about muscle fiber types, and then you're going to be able to describe what differentiates a type 1 fiber from a type 2 fiber, and what the benefit of possessing a high percentage of type 1 fibers is, or a high percentage of type 2. So yeah, that's your assignment after you're able to, or you'll be able to complete this after watching this lecture. And I will go over in detail all of those. So hopefully we will be back in class on Monday and we can do part 3, and then I'm hoping to have one more day for this lecture where we kind of just do a big review of everything, because learning online is sometimes hard for people. So anyway, let's just get started. Get started with our part 2. Okay. So sliding filament theory, muscle proprioceptors, and fiber types. Okay, first we're going to learn about sliding filament theory. This is, I guess, number 3 question on your assignment. So first I'll just kind of briefly explain. It refers to muscle contraction, this theory, muscle contraction and force generation. Muscle contraction actually remained a mystery until an interesting theory was proposed by 2 groups of scientists in the mid-20th century. Sliding filament theory was proposed by different groups, and this theory was created in 1954, and it was published in 2 scientific papers in the journal called Nature, which you really don't have to know about that. But those articles provided experimental evidence revealing how the muscle shortens and develops force. This is the most insightful explanation of how muscle proteins interact and result in force production. And so that's what we're going to learn about. So just kind of an overview, what happens here is that the filaments are going to slide over each other to produce force without changing in length. So what filaments are we talking about? We're talking about actin and myosin. So actin and myosin they're the contractile proteins. They allow contraction to occur. So this sliding process produces the change in the striation pattern. So remember in part 1 we looked at the sarcomeres under polarized light, and it looked like kind of the striated pattern. We'd see these kind of stripes. That's due to the sliding of actin over myosin. That's where the striation comes from, the sliding. So another kind of big point to understand is the number of actin-myosin complexes formed will dictate how much force is produced. So what's an actin-myosin complex, also called an actomyosin complex? It's when actin and myosin interact. And the number of interactions, the number of complexes that are formed, that determines how much force is produced in the muscle. Remember when we said that the myosin heads, 2 of those heads, it's called a cross bridge. That cross bridge has to interact with the active sites on active, and that's going to form an actomyosin complex, resulting in more attachments and greater force production. So yeah, I also want you to understand that the filaments themselves do not change in length. So like actin and myosin, they don't actually shorten themselves, they slide past each other. And remember, myosin is anchored, it doesn't move. It's just the actin that slides. let's look at this picture at the bottom. And I'm really sorry, on my slide or on my video that I did, it's cut off at the very bottom, but you can look at if you have your lecture printed out or if you have it on your computer, you can see it on Blackboard, but it shows the myosin. So you see those green heads there at the very bottom. Well, you should see the entire myosin filament on your slides. But we can get a kind of a good idea what we're looking at here. So this is called the steric blocking method. This is during rest. So see here, this is during a rested phase. So during the rested phase, actin and myosin are in very close proximity, but they're not actually touching. So they're not actually touching. This is the steric blocking method. It's the essential displacement of tropomyosin, and that's triggered by an increase in muscle fiber calcium concentration. In order for us to have that interaction, we have to have calcium. And what do you see up here? We see these black dots. That's representing calcium. You see there, Ca++. That's calcium. So you see calcium is going to bind to troponin C, and that's going to cause the shift in tropomyosin, the long chain, off of the active binding sites. And then that allows the myosin head or the myosin cross bridge to attach on to the active site on actin. So yeah, we're going to learn about this theory, or not, yeah, it's a theory. We're going to learn about this theory in detail. Oh yeah, that's rested phase. You already got that. Okay, so resting phase, what is going on here? Little calcium is in the myofibril. It's stored in the sarcoplasmic reticulum. We defined that term earlier. We defined what the sarcoplasmic reticulum is. It's just a little network of tubules that stores calcium inside the myofibril. And remember, myofibrils are smaller components inside a muscle fiber. Okay. So second phase. So rested phase, there's no interaction. There's no actin and myosin aren't touching. It's that steric blocking method that's going on. And tropomyosin is blocking the active sites on actin. Now the excitation, contraction, coupling phase. So here, an electrical impulse, which is an action potential, generated and spreads across the muscle membrane, which we refer to as the sarcolemma. So it's going to spread across the sarcolemma, which we said has a very nice chemical environment. If you know anything about action potentials from AMP1 or AMP2, you know that action potentials, there's an exchange of sodium and potassium back and forth across the neuron or across the cell, basically. And we will learn all about that in Chapter 5, but I'm just giving you kind of a brief intro to that. We need a nice chemical environment, and the sarcolemma gives us that. It allows for this exchange of sodium and potassium because that's what happens when an electrical impulse is passed. And it spreads across the sarcolemma, and then the action potential will propagate into the transverse tubules and penetrate the sarcoplasmic reticulum. So remember, we said a transverse tubule is a little invagination or a little tunnel inside the muscle fiber, and that's where the action potential propagates or kind of dives down into. And when it does this, it penetrates the sarcoplasmic reticulum. We know this is where all the calcium is stored. So what happens here? A DHP receptor, it's just a type of receptor. It's a receptor, so basically the impulse travels down the T-tubules and penetrates the DHP receptor, which acts as a voltage sensor. So it's like a voltage sensor. So once it's activated, it will then trigger the ryanodine receptor. The ryanodine receptors are located in the membrane of the sarcoplasmic reticulum, and they open once stimulated, and then they release calcium into the cytosol of the cell. So we have two kind of triggers, two receptors that have to be triggered. So sarcoplasmic, or the action potential, is in the sarcoplasmic reticulum, and that's where the DHP receptor is located. And once that voltage sensor is activated, that will then trigger the ryanodine receptor. The ryanodine receptor is activated, and it's going to cause opening of the sarcoplasmic reticulum once it's stimulated, and now calcium is released. So we already know that calcium will bind to troponin C. Troponin C is the only troponin subunit that can attract calcium. So once it's attracted to the troponin C, it binds onto it, it holds tight, and then tropomyosin will shift off of the actin binding sites, those binding sites that are kind of all across the actin filament. Tropomyosin will shift, it will move. And then the myosin crossbridge head will bind to the actin, the active site on actin, and it pulls it. So if you remember, the myosin head, it's on a hinge. It's on a hinged pivot. And so when it attaches to actin, it's going to pull it. It's going to pull it towards the M line, towards the middle of the sarcomere. That is how force is produced. This is called the power stroke. The power stroke, it's kind of described as like a ratchet movement where the myosin head swivels on that hinge pivot and then pulls the actin filament over the myosin filament. And then the sarcomere shortens, which causes the entire muscle to shorten. But this movement has to happen over and over and over again. So we're going to watch a video later that shows how this actually works. So we'll watch a few videos in this lecture. But let me explain it to you with a picture because it's really easy to see it this way. Okay, so the gray is the sarcolemma. So this entire image here is one muscle fiber. So it's one single muscle fiber, and the gray is the sarcolemma, okay? So what happens is the action potential is basically, we have nerves all through our body, and that's how action potentials are passed, or a nervous impulse. And so it's passed to a nerve that's closest to this muscle fiber. And then once it gets close to it, that nerve or that action potential is going to pass across or get into the muscle fiber. So this is the fiber, and it's going to basically slide across the sarcolemma until it transmits down into the transverse tubule. So it's going to slide across it until it hits the transverse tubule, which are these little gray tunnels here. And then once it hits the transverse tubule, it will then activate the DHP receptor, which is located in the blue area, which is the sarcoplasmic reticulum. So after the DHP receptor is activated, then the ryanodine receptor will be activated. And the sarcoplasmic reticulum, which stores calcium, will then release it. And then all of the calcium will flood into the myofibril. So you see right here, there's myofibrils. And this here is a sarcomere. These are individual sarcomeres. And then they're going to attach to the actin, or the troponin C, and shift tropomyosin. So yeah, that's how it works. Okay, here's another picture. I just want to give you as many pictures as you can so you can understand this. So don't worry about number 1 up here because we haven't learned about this process yet. We'll learn about this process in chapter 5. But what you just have to start with is number 2. So the action potential. It propagates into the transverse tubule, and see how all the calcium is stored here? Well, we have these calcium ion channels. So these ion channels will open, and that allows calcium to pass through. And then calcium will bind to troponin, causing a shift in tropomyosin. And then we see down here, calcium binds to the troponin, troponin C, it shifts it. And then the myosin head, which is right here, can attach to the active site, and it can pull actin, and it's going to pull it towards the center of the sarcomere. So yeah, that is another just kind of image to look at how it happens. And remember, that is called the power stroke. The power stroke is when the myosin head attaches to the active site on actin and swivels on its pivoted head and pulls it closer towards the M line, shortening the sarcomere. That's the power stroke. And then when we are done with everything, so let's say that contraction terminates, it's over, calcium is just going to be pumped back into it. So there's these pumps, and we'll learn about it later, but all the calcium that was left over that didn't get used, it doesn't get wasted away. It just gets pumped back into the sarcoplasmic reticulum, and it can be stored there again. One more time looking at this is the power stroke here. So we're about to learn, how does this myosin head become energized? We're about to learn that because it doesn't quite make sense yet, but we'll get there. So it's going to be an energized myosin head, and it attaches to the active site right here. And then we go to this next picture, and it's going, so it attaches to it, and then here it pulls it. And so when it pulls it, it's moving the actin, so the actin's moving closer to the M line. This process repeats over and over and over again. And this right here is the power stroke. Okay, so let's talk about this contraction phase here. So cross-bridge flexion. Flexion is the pivoting, the swivel of the myosin head. It's the pivoting, hold on. Guys. Sorry. Okay. Sorry. Contraction phase. Cross-bridge flexion is caused by ATP breakdown to ADP. Y'all, do you remember what I said the myosin head was made of? Hold on a second. Oh my gosh. Okay. Remember what I said the myosin head was made of? It's made of an enzyme. It's made of myosin ATPase. What does that enzyme do? It hydrolyzes ATP. So it needs energy. So the cross-bridge flexion, in order for myosin head to pivot back and forth, we have to have ATP, which is energy. So when the ATP binds to the myosin head, it breaks it down into ADP, but it releases energy, allowing the myosin head to move. And then a second ATP will replace this ADP right here, and the process happens over and over again. That allows this part to release from it. So then the myosin head will detach from the site, recock, and then reattach again. And so this contraction phase is just how the whole process happens over and over again. So one more time, the myosin head is made of an enzyme, myosin ATPase, that hydrolyzes ATP. It breaks it down into ADP, releasing energy. But then it's stuck there. It's stuck attached to actin. So what happens? A second ATP will replace ADP, and then the myosin head can detach, and the process happens over and over again. And now relaxation. Calcium is just pumped back into the sarcoplasmic reticulum, which is SR, and then actin and myosin return to their unbound state. So I have this picture down here, but we'll watch this little video because I think it's super helpful at looking at, this right here. So if you see this, this is how the process works basically. We see the yellow or the orange, that is the actin filament. The blue is the tropomyosin, and the pink is the troponin. And then we see calcium, you can't really see it, but calcium will bind onto it and shift it. And then this myosin head will pivot back and forth and pull the sarcomere, pull the actin filament closer. So what's flying at the myosin head? That's ATP. So as ATP attaches to it, it hydrolyzes it, and then another one replaces it. So this is the process, which I think is kind of cool to look at. Okay, so one more time, and then we're going to watch a few more videos. We already said this, we already said this earlier. The number of cross bridges bound to actin dictates how much force is produced. So it just depends, it basically determines force production. The more cross bridges we have in the muscle, the more actomyosin complex is formed, the more force we can produce. All right, so we start up here with number 1. The myosin cross bridge attaches to the actin filament. Okay, we see it here. High energy configuration, it's generated. ATP was basically attached to the head, which it then was broken down. And so this is where the phosphate here is used. And then we see number 2, this is the working stroke, or called the power stroke, okay? And so the ADP and phosphate are then kind of released from it after the myosin head pivots, okay? But then the head is stuck there. It's stuck there until we have another ATP over here go over and break it free. So the new ATP attaches to the myosin head right here. So now we have a low energy configuration, but look what happens. The ATP binds to it. Then we have ATP hydrolysis. Now we know energy is released and the cocking of that head can occur. We have ADP and phosphate, and then it happens all over again. So this is the process. Just means that it's energy dependent. It needs ATP to function. But yeah, let's watch a video, and that may help things a little bit. I have 2 videos at the bottom, and I actually don't know which one is the one I want to watch. But let me just click on one, and we can try it out. Okay, hold on a sec. There should be volume.
You use muscles every day to do activities. This woman is using muscles to circulate blood and move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the action between the filaments is described as sliding, the myosin filament actually pulls the actin along its length. The cross bridges of the myosin filaments attach to the actin filaments and exert force on them to move. This action is known as the sliding filament mechanism of muscle contraction. In this model, the sarcomeres shorten without the thick or thin filaments changing in length. A contraction begins when a bound ATP is hydrolyzed to ADP and inorganic phosphate. This causes the myosin head to extend and can attach to a binding site on actin, forming a cross bridge. An action called the power stroke is triggered, allowing myosin to pull the actin filament toward the M line, thereby shortening the sarcomere. ADP and inorganic phosphate are released during the power stroke. The myosin remains attached to actin until a new molecule of ATP binds, freeing the myosin to either go through another cycle of binding and more contraction, or remain unattached to allow the muscle to relax. Muscle contractions are controlled by the actions of calcium. The thin actin filaments are associated with regulatory proteins called troponin and tropomyosin. When a muscle is relaxed, tropomyosin blocks the cross-bridge binding sites on actin. When calcium ion levels are high enough and ATP is present, calcium ions bind to the troponin, which displaces tropomyosin, exposing the myosin binding sites on actin. This allows myosin to attach to a binding site on actin, forming a cross bridge. Calcium ions are stored in the sarcoplasmic reticulum and are released in response to signals from the nervous system to contract. Neurotransmitter molecules are released from a neuron and bind to receptors, which depolarizes the membrane of the muscle fiber. The electrical impulse travels down the T tubules and opens calcium stores. Calcium ions flow to the myofibrils, where they trigger a muscle contraction. As the actin and myosin slide along each other, the entire sarcomere shortens as the Z lines draw closer to the M line. As the sarcomeres in myofibrils contract, the entire muscle fiber will shorten. When muscle fibers contract in unison, a muscle can produce enough force to move the body, allowing you to take notes.
Okay, hold on. Let me just get out of here.
I have to stop.
Okay, Sorry, I kind of got lost where I was. I started lecturing just now and then realized I wasn't even recording. So excuse me if I left off somewhere. But I'm going to start with this slide. Okay, so proprioception. What is proprioception? This is how the body senses where it is in space. In the sense of our neighboring body parts. So it's just being able to know where your arms, legs are without having to look and see where they are. You just know where they are. That's what our proprioceptors are for. Think about how important that is during a sport. When you're running or kicking or throwing or hitting something, you need to know where exactly your arm or leg is positioned in order for a successful sport performance. And running, basically any type of movement we do, we need to know where our body parts are so we can perform optimally. And that's what our proprioceptors are for. This is also called kinesthetic sense. Kinesthetic sense is the brain's integration of proprioceptive inputs via receptors located in muscles and tendons. Basically, the information is sent to our conscious and subconscious portions of the brain. This allows for a repeated complex skill to just feel right. This requires successful practice, though, to learn a motor pattern and to reproduce it over and over again. So then we can conduct it so quickly that we don't even have to think about it. So that's kind of what proprioception is for, and I'll go over that one more time in a second. But let's talk about some proprioceptors in our body. So we have them located in our entire body, but the only ones we care about and that we're going to talk about are the GTOs, so Golgi tendon organs we see down at the bottom, and then muscle spindles. So those are going to be the two that we learn about. But they're located in our muscles and our tendons. So the GTO is in the tendon and muscle spindles obviously in the muscle. But what does this allow for? The learning effect, which I just kind of explained. It's the ability to repeat a specific motor recruitment pattern results in successful performance of a skill. So this is important, especially when we are learning new skills. This keeps the central nervous system constantly informed as the body is learning a new movement subconsciously. So it allows the learning effect. It's basically when we repeat a skill over and over again and we're repeating it correctly, it's kind of integrated into these proprioceptors. So we're able to just learn it and perform it. This allows for perfect performance the more we learn and practice something perfectly. So like I said, important in learning a pattern skill when position, length of a muscle, that's important. And then when like force, tension, all of that. So again, it just requires successful practice to learn a motor pattern, reproduce it over and over. So by the end of all of that we don't even have to think about it because we have done it so much that our proprioceptors can remember exactly how to do it and we can perform something. So yeah, that's what proprioception is. We are going to talk now about muscle spindles. So muscle spindles are a gamma motor neuron, and we'll talk about differences between different types of neurons. We've got gamma motor neurons, we have alpha motor neurons, so we'll kind of learn about the difference between those. But let's talk just about what does the spindle do. So it has two functions. It's going to monitor length and stretch of a muscle and rate of stretch. So that's what it's monitoring. And then it will initiate a contraction when the muscle is stretched. So let's follow these steps down here. So stretching of muscle stimulates a muscle spindle. So a muscle spindle, it's an interphysial fiber, which means it's embedded inside the muscle. So if you look here, this is muscle tissue, and then these are the interphysial fibers. So interphysial fibers that are surrounded by connective tissue. So I'm going to go over these. There are different types of fibers. There's one dynamic nuclear bag fiber, one static nuclear bag fiber, and 5 nuclear chain fibers. All are composed of stretch-sensitive tissue. So what happens? So the stretching of a muscle, you see it's being pulled, it's going to stimulate the spindles, and then the spindles are connected to, they're basically this gamma motor neuron here. And this gamma motor neuron is going to send information, sensory information, to the spinal cord. And so here, this is a cross-sectional view of the spinal cord. It just means you take the spinal cord, you cut it in half, and that's what you're looking at. You're looking at the inside of it. So this is kind of where a lot of really fast reaction type movements are produced. Or maybe not produced, but controlled. So yeah, this is what happens. So it's going to, sensory neuron is going to attach over here. It's going to be, this information is going to be processed. And then the motor neuron is going to send it back to, so sending information back to the muscle that was stretched, but it does contract the muscle. So that's what happens. It monitors length and stretch, rate of stretch, and initiates a contraction. So muscle spindles are going to be active anytime a muscle is stretched. I also would do this if y'all were in class with me. I would make you stand up right now and touch your toes again. And you would feel that stretch in your hamstrings. So I'd make you go down really slow, and you would feel your hamstrings getting tighter and tighter. So your muscle spindles are firing, and that tightness you feel your muscles start to contract a little bit instead of relax the more you stretch that muscle. The purpose of that is to prevent overstretching. And so I would also have you're still standing up. So if you're still standing up, I would have you touch your toes quickly. And when you touch your toes quickly, your muscle feels tighter faster. That's called the stretch reflex. And the stretch reflex is when a muscle is quickly stretched, it initiates an immediate contraction. So the contraction's initiated immediately. And yeah, so we said that tighten also contributes to this. So tighten and muscle spindles contribute to the stretch reflex, where we have contributes to passive stiffness in the muscle and limits range of motion. So that's what the stretch reflex is. It just prevents overstretching of the muscle. Muscle spindles are active in all sports and exercises that involve a degree of muscle elongation. So any movement that you do in your exercise where muscles are elongating and they're stretching, maybe stretching quickly, muscle spindles will be active. So maybe get creative when you answer your question on your assignment when it's asking about muscle spindles and during exercise. So think of sports where the muscle is stretched quickly. Any muscle stretched quickly, muscle spindles will be stimulated. So let's watch this little video here about muscle spindles.
Proprioception is imperative in the body's ability to retain balance and posture without conscious effort. In order for the human body to orient itself in space, special sensory receptors called muscle spindles are embedded in the belly of skeletal muscle. These receptors detect changes in length and velocity of stretch. Typical muscle spindles consist of 3 types of intrafusal muscle fibers, surrounded by a connective tissue sheath. These include 1 dynamic nuclear bag fiber, 1 static nuclear bag fiber, and around 5 nuclear chain fibers. As action potentials reach the muscle via alpha motor neurons, the surrounding extrafusal fibers contract or relax, stretching the muscle spindle along with them. Sensory information on the velocity of this stretch and muscle length is transmitted via afferent sensory neurons. These signals are transmitted to the central nervous system and modulated by efferent gamma motor neurons that regulate the contraction of the muscle spindle fibers. This coactivation of muscle fibers allows the body to sense and maintain its position in space. Without it, balance becomes a near impossible task.
Okay, so you kind of see what the role of the spindle is. Now we have to talk about the role of the GTO, the Golgi tendon organ. So GTOs are going to be a little different. These are extrafusal fibers. Extrafusal mean they're outside of the muscle. They're located in the tendon, as you see located down here. They're located near the myotendinous junction. So it's just like right in between where the muscle belly and the tendon meet the bone. So just kind of in between there. So what does the GTO do? Main function is it monitors and responds to tension. So they fire, so they're activated with excessive force on the tendon. And then their response is to relax the muscle. So the opposite of the muscle spindle. So, okay, let me just kind of start. Let's do an example. So, well, actually, first, let me just show the process. So we see GTOs down here, and then the muscle spindles on the inside. So let's say there's a lot of tension or force placed on a joint. Well, then the GTO is going to fire, and it sends the sensory neuron to the spinal cord, and it's then going to be processed in the spinal cord, and then it sends a motor neuron back to that same muscle, and that's going to cause the muscle to relax. Because the muscle's already tight, there's a lot of tension on it, there's tension placed on the body, it causes the muscle to relax. So why does it do this? It basically is it's a protective mechanism. It's like an innate protective mechanism that was like ingrained in us as humans. And the too much tension, if we try to contract against too much tension, then that may hurt the muscle or injure the muscle. So the body's natural response is to relax the muscle. But resistance training seeks to inhibit the action of the GTO to allow for greater force production. So normally I would turn this question to the class and say, how does heavy exercise affect proprioceptive sense? How does it affect that? So what we would basically say here, well, first let me go back to this bullet point right here. So resistance training. I would normally ask this question. Okay, so let's say we have a heavy barbell on our back and we're doing back squat. So we're doing back squat. We've got a barbell on our back and we're going down. We're lowering. Okay, think about our joints. Every joint under the barbell and every muscle under the barbell, there's tension placed on it, right? So all of that tension being placed on our body, that's going to fire these GTOs. These GTOs are going to fire because they're responding to a lot of tension or pressure or force. And so what happens? The body is supposed to relax. Well, I would normally ask this. What would happen if we let our body relax with a heavy load on our back? We would fall to the ground, right? I know somebody was thinking that. We would fall to the ground. You're right. So resistance training actually wants to inhibit the action, which means to stop the action of the GTO to allow for greater force production. Because what's the purpose of resistance training? It's contracting against a heavy load. It's creating force against something heavy, against a heavy load. So yeah, the more resistance training we do, the more we can actually inhibit the GTO. And if we can inhibit the GTO, we can actually produce greater force. Because the GTO is trying to relax the muscle when it feels a lot of tension. We don't want that. We want to contract against all the force. So resistance training inhibits the action of the GTO, and we are more forceful. You need to remember this. We are going to talk more about this in chapter 5, so you need to understand this now. Okay, second question, or second part. How does heavy exercise affect our proprioceptive sense? So we know proprioceptive sense of the body's position is affected by fatigue. Like you would assume, like our body gets tired, and with any onset of fatigue, our proprioceptive sense kind of slows down and becomes delayed. So we want to understand that the importance of training and conditioning is to reduce fatigue during exercise. This allows, or basically the goal is to enhance the flow of information from muscles back to the central nervous system, and then back to the body. So with heavy exercise, sorry. Okay, so with heavy exercise, our proprioceptive sense becomes slowed down, delayed due to fatigue. So just like you would think when our muscles get tired, so does our central nervous system. And this is part of the central nervous system a little bit because we're looking at how it communicates with the spinal cord and sends information back to the body. So with heavy exercise, It's going to be affected by fatigue, which means these signals may slow down a little bit, which means we don't get information quick enough, which sometimes can result in injury or poor performance. So yeah. Okay, that's all about muscle spindles and GTOs. So you've got enough information now to answer number 4 on your assignment. So for number 4, I asked you, what is proprioception? So just explain, give me a sentence or two explaining proprioception, and then explain the difference between muscle spindles and GTOs. But also you have to tell me about during exercise. So remember with spindles, think of whenever the muscle is stretched, what exercises or sports do we stretch our muscles, or when are they moved or elongated? And then with GTOs, you need to think about when is there a lot of tension or force placed on the body. And that's how you can answer the exercise question about GTOs. But okay, we are on the last kind of major topic, which is probably the best topic. It's muscle fiber types. This is one of my favorite topics. It's really neat. So yeah, let's get started with this. And remember, this is number 5 on your assignment, and then you'll be able to complete it. Okay, so skeletal muscle is a mixture of 3 major fiber types. There are more than 3 fiber types, but we are just going to talk about 3 major fiber types. We have slow twitch and we have fast twitch. So our slow twitch fibers are called type 1, and then SO stands for slow oxidative. And then fast twitch, we have type 2A, which is the FOG. F stands for fast, O stands for the oxidative, same thing, and G stands for glycolytic, which means glycolysis. So oxidative literally means aerobic metabolism, and glycolytic means glycolysis. And then type 2X is just fast glycolysis. So they are all different. Type 2A is an intermediate fiber. It's basically a mix between type 1 and type 2X. It's an intermediate fiber, a mix between the 2 kind of extreme fibers. So yeah, what else can I give you before I move on? We're going to talk about different classifications that were developed on the basis of histochemical and biomechanical and physical characteristics, which are going to be talked about in just a second. But I do want you to know that fiber type is genetically predetermined. Fiber type is established shortly after birth, and it's genetic. So it's based on our mom and dad. So our parents is where we get our fiber type from. So we can get maybe a mixture of both parents, or we could be maybe more mixed with what our dad had or more with what our mom had. But yeah, it is genetically predetermined. We can't do anything about it. Okay, what are the classification systems of fiber types? So first, I want you to look under here. This is muscle fiber under polarized light. This shows a type 1, this shows a type 2A, and this shows a type 2X. Just know it used to be called a type 2B. In research years and years ago, but recently they have changed that. So if you ever see a type 2B fiber like on an old book or an old image, it's the same thing as a type 2X. Okay, how are fiber types classified? So 1 is type, which I already went over. So type 1 versus type 2 fibers. Speed, we already talked about as well. Slow twitch versus fast twitch, and it literally means their twitch speed. So have you ever had a muscle twitch before? Like think about it, if your muscle like twitches or spazzes a little bit. So that's like a twitch speed. So it depends on how quickly does the muscle twitch. And that can result in how quickly it can produce force or how quickly it can respond to force or movement. Next is metabolism, we also already talked about. So slow twitch fibers rely on oxidative metabolism. That's what the O is, metabolism. And then fast twitch, our type 2A relies on oxidative and glycolytic, so it can be fast or slow. And then the type 2X is just glycolytic, glycolysis. Another is color. So with color, we have red fibers versus white fibers, but I do want you to know that this picture is not what the color of what they actually look like. Those fibers are stained. They're stained with like a dye solution. So those aren't actually what they look like. What they actually look like is a type 1 fiber is red in color, It's got this reddish color to it, and we'll talk about why, but I'll talk about why actually on the next slide. I'll get to it, but it's red in color. And then the type 1 is really, the type 2X is really white in color. It's white, but we'll talk why. Okay, so let's just go into more characteristics. We have to know the difference between the two. So we already know type 1 is slow twitch. Slow twitch means it's slow to reach peak force. We can look at the graph down here and we see, you can't see the x-axis, but it's over time. So over time measured in milliseconds. Look at the red line compared to the blue line. And this is like with activation. So like if we activate one of the fibers, the type 2, this fast twitch, produces force really quickly in a short amount of time, whereas the slow twitch does not produce force as quickly. And it doesn't produce as much of it. So it's slow to reach peak force and low total force production. So we can't produce a lot of force with type 1s. They are not very strong fibers, but they do have a high capacity for oxidative metabolism, which means they rely on aerobic metabolism for energy. So let's talk about why that is. Why are they oxidative? Type 1 fibers have greater capillary densities. So the capillary beds inside a type 1 fiber, there's more capillaries, which means more blood flow, which means more oxygen delivery. And they have a high myoglobin content. We know that myoglobin is a cytoplasmic hemoprotein that reversibly binds oxygen inside of the cells, which a muscle cell is the same thing as a muscle fiber. So more myoglobin, which transports oxygen to the mitochondria. So if there's more myoglobin, we can transport oxygen faster. And there's also more mitochondria. So more mitochondria, which means more ATP, which means we can exercise for a long time, but at a low intensity using type 1 fibers. They are also fatigue resistant. They contract over long periods of time at low intensity, and they are not fatigable, which means it takes a lot of effort to fatigue them. But remember, that intensity has to be low. So based on all of those characteristics, I would turn to y'all and say, okay, during which type of activities do we rely on type 1 fibers the most? And you would probably say aerobic sports or aerobic endurance events. So low force activities that requires less fatigue over time. So like walking. Walking is a great example of when we would use type 1 fibers. Also running a marathon. We've got to do that for a long time. And so think of marathon, triathlon, long duration cycling event, long distance walk, cross country skiing, long distance swimming, anything where we're doing something at a low intensity for a long period of time. They're fatigue resistant, which is great. We can use these fibers for a while before they get tired. Okay, and now let's talk about type 2 fibers. So type 2 fibers, and I'm going to talk about type 2 fibers as a whole. So I'm going to talk about both type 2X and type 2A. They produce force rapidly. So once activated, they will contract very quickly, and they contract quickly and they produce a lot of force. So high force production and rapid response rate. So a fast twitch rate. They produce ATP rapidly, but at a very low capacity. So low capacity for oxidative metabolism, but a high capacity for anaerobic metabolism. Remember, they're glycolytic fibers. They rely on glycolysis for energy, to move, to contract. So low oxidative, but high anaerobic. They produce force quickly. They fatigue easily, though. So they fatigue very quickly. They get tired fast. They run out of energy, speed, and they can no longer contract. So now I would turn to y'all and be like, okay, so based on what I just said, during which activities do we rely on type 2 fibers the most? You would probably say You'd probably say resistance training. You're correct. You would probably say sprints. You'd probably say plyometrics. You'd probably say a lot of sports where I have to sprint or jump or kick or do anything rapidly. So any rapid movement or any movement that produces a high amount of force, that's when we rely on type 2 fibers. Okay, so let's look at the bottom chart down here. We want to just kind of compare all three of them. So over here, let's start with type 1, slow oxidative. So slow twitch speed, small amount of force, but high resistance to fatigue. And then type 2A, fast twitch speed, medium twitch force, and also a high resistance to fatigue because the FOG, they have more oxidative enzymes and they use oxidative metabolism more so than the type 2X. Remember, this right here says type 2B, same thing as the type 2X. Fast glycolytic. They're fast, they produce a large amount of force, but they fatigue out very quickly. So yeah, those are the difference between the three fibers that you have to know. This chart here is a really great resource because it shows all of kind of the characteristics that I already went over and looks at type 1 versus type 2. Remember, the type 2A is a type 2 fiber, so it falls in this category, but it does take on characteristics of a type 1. So just keep that in mind. But like I said, this is a great chart to look back at and study, to use on your assignment. It's just a great chart. Let's watch a YouTube video. Not.
You're always earning with Chase Freedom Unlimited.
Sorry.
Writing's not that easy.
Not this either.
But Grammarly can help. This sentence is grammatically correct.
This video.
Muscles are the driving force of all movements in the body, varying from lifting, running, walking, and even organ function. The muscular system consists of three major muscle types: cardiac muscles, which are responsible for function of your heart, smooth muscles, which are responsible for the function of all your other organs, and skeletal muscles, which are responsible for, as the name suggests, the movement of your bones. Both smooth and cardiac muscles function involuntarily, meaning they operate by themselves. Skeletal muscles, on the other hand, function voluntarily and are under our conscious control. There are three different skeletal muscle fiber types, known as type 1, type 2A, and type 2X fibers. The difference of size, color, contractual speed, contractual force, and energy source classifies each fiber. Type 1 fibers, also known as slow twitch fibers, are the smallest fiber types with a darkish red color. It has a fairly slow twitch speed and produces a relatively small amount of force when contracted. It has high amounts of mitochondria, which are organelles within each cell that uses oxygen to produce energy. Although the force generated is fairly small, type 1 fibers are highly fatigue resistant, allowing it to be active for long periods of time. They are the primary fibers used during low-intensity activities with steady oxygen consumption, such as walking, jogging, or aerobics. Type 2A fibers, aka moderate fast twitch fibers, are also red but intermediate in size. These larger fibers typically use a combination of oxygen and glucose as a source of energy. This combination allows for quicker contracting speed and higher force output compared to type 1 fibers. However, fatigue resistance isn't as high. These fibers are typically activated during aerobic activities that are moderate in duration, such as a mile run, swimming, and short-distance cycling. Type 2X fibers, aka fast twitch fibers, are white in color due to the low oxygen capacity and by far the largest fiber type. It makes up for the lack of oxygen capacity by having extremely high levels of glucose in its stored form of glycogen, producing the fastest twitch speeds and the most force. The downside, though, is that the fiber fatigues quickly, burning out after 15 to 30 seconds. High-impact, heavy resistance activities such as lifting weights and sprinting will activate type 2X fibers the most. For any activity, your muscles follow a certain recruiting order. The slow twitch, low force, fatigue-resistant type 1 fibers are always activated first. When type 1 fibers are maxed out, type 2A fibers are activated, and then after those are maxed out, type 2X fibers are activated. This order, known as the Henneman's size principle, helps minimize muscle fatigue and allows precise motor control by using no more than the force necessary to complete a movement.
Okay, sorry about my pups. They hate cars. Any single car that drives by. But you can finish that video if you want to, but it just gives you more information about the fibers. I've highlighted some boxes here that I think are going to be most important for you to learn. just kind of most important to understand. But yeah, learning everything is going to be great, but the ones I highlighted are going to be the best, which I found kind of interesting, the difference between glycogen stores and triglyceride stores. So there's no difference between glycogen stores in type 1 and type 2. They can both store the same amount of glycogen, but look at fat stores. Fat stores are higher in our type 1 fibers, but think about it, they use oxidative metabolism, which can metabolize fat. So of course, makes sense that fat is going to be higher in type 1 fibers if it's an oxidative fiber. But type 2 has a low amount of fat stored in it because it only uses glycolysis, which is pretty interesting. And then look up here at ATP stores. So type 1 fibers store a low amount of ATP, but they do have a high amount of mitochondria, which allows us to produce more ATP. And then type 2 fibers already have a lot of ATP and phosphocreatine stored in it. But yeah, pretty interesting, pretty cool. Okay, let's talk about how do we determine fiber type. So, yeah, let's talk about this process here. So this process is fiber staining, basically, but they did a muscle biopsy. And I should probably show y'all a muscle biopsy. I have a really good video that I need to show, and I may show it next week since we have talked about it and you haven't seen one. But it's basically where they stick a small needle inside the muscle and then they extract a chunk of muscle tissue and they pull out your muscle fibers. And then they would cut those muscle fibers into small pieces. They would slice them cross-sectionally. And then they would stain them, and that's what kind of we're looking at right now. So let me just kind of start talking about this, but where to start? So type 2 fibers have faster form of myosin ATPase, so ATP is split more rapidly. What does this staining procedure do? Y'all remember the enzyme myosin ATPase? It is the enzyme that is on top of the myosin head. So that enzyme has different isoforms where some result in quicker enzymatic reactions for ATP hydrolysis or ATP breakdown, thus higher cycling rates for those fibers for actin-myosin interaction. So basically, actin and myosin can interact quicker and faster, resulting in faster contractions. So that's directly related to the speed at which the myosin head binds to actin and swivels to generate force. So it produces functional classification based on muscle fiber shortening velocity. Finn, stop it. Okay, so what are we looking at here? You can see the second part's cut off, but we have a pH of 4.3 and 4.6. That's going to be our acid. I don't know if I have this. No, I don't. That's going to be our acid bath, Finn. And then we have an alkaline bath, which is the pH of 10. So difference between alkaline and acid bath, you probably know is there's like bases and there's acids. So it's just kind of looking at pH level. We know that as acidity increases, pH level goes down. So here we have a low pH and here we have a high pH. So it's just staining them with just different types of acidic solutions. It's a little above my area of knowledge, so I'm going to try to keep it as simple as possible. Okay, so this is histochemical staining. Results in different stain intensities among the fiber types, and then we can divide them into different categories and assign them to a specific type. And it gives these different unique shades of gray that we're looking at there. So like I said, or remember I said earlier, we have more than just 3 fibers, but I'm only talking about 3 fibers. You can see we have type 1, type 1C, type 2C, type 2A, type 2AX, and type 2X. So you know that we are only concerned with type 1, type 2A, and type 2X. Those are the only ones that we are concerned with. So this is the staining procedure. They're just staining it with enzymes. They're staining it with a solution that has enzymes in it, and then it'll like get really dark if it has a lot of that enzyme. So if we look here at this one, and these are going to be A, B, and C are different pictures, and I don't know exactly what they're talking about, but we can also stain the fibers with a oxidative bath. So this myosin ATPase, this would be a solution that would have a high affinity for anaerobic enzymes in then if we did an oxidative bath, it would be a high affinity for aerobic enzymes. So that would just allow us to identify all the type 1 fibers versus all the type 2 fibers. And so you can just see here, just based on the different baths that they stain them with, they're going to respond differently. Again, like I said, this is a little above my area of expertise, but I will stop there with talking about staining procedures. But that's how it's done. One topic I really like talking about is how, I guess, percentage of fiber type and how we can train our fiber types to act a little differently. So this is a really cool topic. So on average, I have written down up here roughly percent fiber type. So in this, when we think about on average, you can't think of like all your athletic friends or all your friends who aren't athletes and just sit around all day. You have to think of the average person. So someone like in the middle, someone in between that. So the average person, most muscles composed about half type 1 and 25 type 2A and 25 type 2X. So basically 50-50. So it's like 50% type 1 and 50% type 2. That's usually what the average person is, their muscles are kind of composed of. Also, one thing to know is the muscle fibers like in your upper body and lower body are usually going to be similar. So it's genetic, which means if we have fiber type in one area, like if you did a muscle biopsy in your lower body and it was like really high type 2, your upper body is probably very similar to that. I'm not saying it's exactly the same. I'm saying it's similar. So yeah, proportions in one muscle compared to another muscle, it's not going to be identical, like percent-wise, but it will be similar. Let's look at this picture here. I find this very fascinating. So sprint runners, we have male and female. Gastrocnemius, that's going to be your large calf muscle. Let's look at our sprinters. Our male sprinters and female sprinters have a roughly around 25% type 1 and 75% roughly type 2. And then we look at our distance runners, males and females. It's just opposite, like completely opposite. 70 to 80% type 1 for our distance runners, fiber percent. And then 20 to 30% type 2. Y'all, that's so fascinating to me. But let's talk about why that is. So I make these jokes sometimes, like, why did our parents put us in like every single sport when we were younger? To figure out what we're good at, right? To figure out, oh man, like they're a really good sprinter, or they're really fast, or they're really strong, you know, then you're probably going to be more of a type 2 athlete and you're going to stick with those type 2 sports like sprinting and high force producing sports. Or let's say your parents put you in a bunch of sports and they're like, you know what, they can't run very fast and they are not very strong and very powerful or very quick, but you know what, they can run forever. They can run for a really long period of time. And then that's what we know that we're good at. So basically the reason we're looking at this, it's not because the sprinters, their fiber types like changed. It's not because they changed. We don't like all start out with 50-50. Remember, that's the average person, but our sprinters and our distance runners probably are not the average person, remember? So if I had like just a random sedentary person here, they might be 50-50. But we look at these athletes here are sprinters, and the sprinting that they are doing did not turn their type 1s into type 2s. That did not happen. And all of our distance runners, their distance running did not turn their type 2s into type 1s. That's not what happened. It's just like they figured out what they were good at, and then they stuck to that. And so most sprinters have a high percent of type 2 fibers, and then most distance athletes have a high percentage of type 1 fibers. But one fiber can transition, and that is our type 2A. Sorry. I'm sorry. I don't even know what I'm talking about. Type 2X fibers have been shown to transition to a type 2A, and that's what this picture down here looks at, this one at the bottom. So what does this show? Exercise activities. So any type of exercise activity, muscle contraction, basically the type 2X will shift to a type 2AX, which will then shift to a type 2A. So this here is kind of what we know, what researchers know. That's why these arrows are bigger. it shows that with any type of muscle contraction repeated over and over and over again, that we're going to see a shift in the fiber from the X to the A with regular exercise. Why is this beneficial? I would normally ask the class. Because why is a type 2A fiber, why is it so special? Because it is both oxidative and glycolytic, which means it can produce force, but it doesn't fatigue as quickly. It has higher fatigue resistance than a type 2X does. So that's why having a high percentage of type 2A fibers is really beneficial. But yeah, so that is one transition that occurs. So this transition occurs because our type 2X fibers are actually kind of inefficient. They're inefficient because they fatigue out too fast, and then we can't use them anymore. So it's beneficial if we can transition those to a type 2A fiber, and means we could sprint for longer, or we can lift heavier for more contractions. So this shift is a very beneficial shift, and it's really happens to anybody who produces continuous muscle contractions over and over and over again, which is very awesome. But yeah, let's talk about why this fiber, let me say one more thing. Type 1 fibers cannot turn into a type 2. When I say cannot, it means research has not figured that out yet. So see this question mark here? We're looking at the transition between a type 2 fiber and a type 1 fiber. Research has not shown that yet. We basically we don't know. We don't know if that can happen. Maybe one day they will figure out that it can, but right now it cannot. So I just want you to know that. Okay, y'all, this is like the last slide, so we're almost done. The myosin molecule, why does a type 2X and a type 2A, why can they switch back and forth but not to a 1? Well, what research knows so far is that the myosin heavy chain content is similar between an X and an A. So let's just kind of look at this. This is the myosin molecule. We have the tail, which is made up of these light chains we see down here. So we have light chains, and then we have the ELC, which is the essential light chain. We have the RLC, regulatory light chain, and then we have the heads up here, and the heads are heavy chains. And so that's what the MHC, and we look at this picture here. The MHC between myosin heavy chain content between a type 2X and a type 2A is very similar. So basically they can transition back and forth, but it cannot transition to a type 1, which we see myosin heavy chain content of a 1, because it's just that content is different. But yeah, this is also how we determine fiber type, is by looking at the myosin heavy chain content. And when we look at this content in the staining procedure, that's what helps us determine fibers. But yeah, this just shows why a type 1 cannot become a type 2 due to the myosin heavy chain composition, whereas a type 2X and an A, they can transition based on physical activity or inactivity. What I didn't say earlier is, which is also really fascinating, let's say you were an athlete in high school and you were a really good athlete and you saw this transition between an X to an A when you were high school and really active, but since you've got to college, maybe you've slowed down your activity like a little bit. You know what happens is this type 2A fiber will actually transition back to a type 2X. But you're thinking, oh wow, like that's beneficial, right? Like I can produce a lot more force. Well, you're not exactly. You just lose your ability to continue that force production for the prolonged force production or prolonged speed. So we see a transition back to this fiber here. The type 2A fiber is very beneficial because it's active and it produces lots of force really quick, but doesn't fatigue as quickly. But that was it. Whew, that was a long part. I go into a lot more detail when I'm on video rather than in class. So this is your Chapter 4 quiz review that you can take a picture of and prepare for that quiz whenever we get back. I think this quiz is on Wednesday for you. So, and then for my Tuesday, Thursday class, if I don't see you, it's going to be whatever day says it in your schedule. But yeah, so, and also my Tuesday, Thursday, your assignment's due Tuesday. For my Monday, Wednesday, Friday, your assignment is due on Monday night. But yeah, that's it. We are done with part 2. Give me just a second. Thank you so much. I will be available to chat via email if you want to shoot me anything. I know I covered a lot of material. Like I said, I go into a lot more detail on videos because I can. And yeah, so I hope you are safe and warm and enjoy your weekend. But reach out to me if you want to. But yeah, I will see y'all hopefully next week.